Continue now to the next set of notes. The next set of notes is about organic chemistry. Um, so you might know a little bit about chemistry because it's a requirement. I think in most high schools that you take a physical science, and even in the life sciences, there are some units that go over chemistry where you talk about maybe atoms and molecules and whatnot. Even in high school biology, we go over that. And you might be familiar with chemistry, just using chemicals in your everyday life to cook, to clean, and that kind of stuff. But when it comes to living things, when it comes to the human body, the chemistry of the human body is very unique. All right. So when we break things down, when we break our tissues down, our organs down, everything really gets broken down into organic molecules, right? Organic molecules. These organic molecules are things like DNA. DNA is a nucleic acid. Lipids. Lipids are fats, oils, waxes, carbohydrates, proteins, things like this. And when we break each of these down, we see that the atoms that are used to build these large organic molecules typically are carbon, oxygen, and hydrogen, and sometimes nitrogen. Nitrogen is found in the DNA and the proteins, not so much in lipids and carbohydrates. So organic molecules are molecules that are made by living things, found in living things, and they each contain carbon, hydrogen, and oxygen. Right? So this is kind of the definition of an organic molecule. So when we break down the molecules that we typically find in the body, proteins, I mean, most of our structures are made from proteins, right? That's one of the most important jobs of the cell is to make proteins. Carbohydrates are something that we use for energy. We store carbohydrates throughout the body in our muscles and our liver and whatnot. We use them for energy. Same thing with fats. DNA is the genetic information that's stored in our cells. But one thing that is common amongst all of them is that they each have a carbon backbone with hydrogens and oxygens, all right? So that's kind of important. But then we also see smaller traces of these items down here in the body: calcium, phosphorus, sodium. We see these things in smaller quantities clinging onto to the molecules of the body. So these things that we're talking about, like carbon, oxygen, and hydrogen, these are referred to as atoms. And we can see them on the periodic table. The periodic table is the table of elements, right? But if we could break down each element into its smallest possible unit and still consider that element to be pure, what we would have is one atom of that element, all right? And what atoms do is, many of them, is they react with each other. They form bonds with each other, all right? And when we do that, we create what are called molecules. So molecules are formed when different atoms like carbon and hydrogen start clinging to each other. If we look at the atom, we could kind of break it down into a couple different parts. We have positively charged particles. These are called protons. We have negatively charged particles. These are called electrons. So the negative ones are called electrons. And as you can imagine, the negative ones and the positive ones are attracted to each other. So typically, when we're looking at the elements in the periodic table and all those atoms, these atoms are neutral. And what that means is that the number of electrons in each of them is going to be equal to the number of protons. But then we have these other ones that are found in the nucleus called neutrons. And we could represent them by just like an empty circle with no charge. And they don't carry a charge, neutrons. They just contribute mass. All right, so we have an electro chemical attraction that's happening between the electrons, the negatively charged electrons, and the positively charged protons. Remember, the protons are going to be found in the nucleus. And this electromagnetic attraction between protons and electrons is what keeps the electrons local. It's what keeps them around. So if we look at this as an image, right? Here's our nucleus. The nucleus of an atom is right here. All the subatomic particles that are found in the middle. And what do we find in here? Well, we find the positively charged protons, and we find the neutrons that don't have any charge. And surrounding this, we have these particles called electrons. This atom has how many electrons? Well, we got 1, 2, 3, 4, 5, 6. 6 electrons. If there are 6 electrons that are negatively charged, if this is an atom from the periodic table, then there has to be 6 positively charged protons. This number has to be the same. All right. This number could be different. It could be anything. This number doesn't matter because it doesn't contribute to the charge of the atom. Right? It just contributes to mass. All right. So just keep in mind that in the nucleus we have protons and neutrons, and surrounding the nucleus we have these negatively charged electrons. This shell out here, and these electrons are moving around. They're kind of spinning around, orbiting the nucleus. And these trails are called orbitals, right? And so in this case, we have 2 orbitals. The first orbital could only have 2 electrons, and that's why we see 2 here. The first orbital cannot have more than 2. But this atom has 6 electrons, so what happens is we have to create a second orbital. In the second orbital, we can have up to 8. But in this case, we only have 4. But this second orbital can hold a maximum of 8. And so there's plenty of room for the 4 remaining electrons to exist in this orbital. So this is the outer shell. This one right here. This is the outer most orbital. So this one has a name. This is called the valence shell. The outermost shell is called the valence shell. And the electrons that are moving around in that outer shell, these are called the valence electrons. Valence electrons are very important because it determines how reactive this atom is going to be. All right. The ones on the inside, these electrons here, these are not valence electrons because they're not occupying the outermost shell. All right. So this atom here has, even though it has 6 electrons total, it has 4 valence electrons. This is a couple different pictures of a couple different atoms. We have oxygen, we have hydrogen, we have carbon, and we have nitrogen. And you can see we have different orbitals. If we look at oxygen, if you look at oxygen on the periodic table, for example, on the periodic table, you would see the letter O for oxygen. And then you would see a number above that says 8. And then you would see a number below that you would round. I think it's on the periodic table, I think it's like 15.99, something like that. But you would round that number to 16. All right. This number at the top is the atomic number. And this number at the bottom is the atomic mass. These numbers mean different things. The atomic mass tells us how many particles are in the nucleus. How many particles are in the middle? Now remember, in the middle, in the nucleus, we only have protons and neutrons that have zero charge. So how many of these particles are we going to find in the middle? And that will give us our atomic mass. Well, it says 16. So you would assume that there's 16 particles in here. This number at the top tells us how many of those 16 particles are going to be protons. So the atomic number is the number of protons. So out of the 16 particles in the nucleus, 8 of them are protons. So that leaves, if you subtract 16 from 8, that leaves 8 of them to be neutrons. And of course, as I said before, all these atoms are neutral. So if there's 8 positively charged particles, we can expect to find 8 negatively charged particles, 2 in the first shell, and then we need 6 more. So this molecule of oxygen has 8 electrons, but it has 6 valence electrons. Right? Even though a total of 8, because this is a neutral. If there's 8 positives, there has to be 8 negatives. But in the outer shell, in the valence shell, we see 6. All right, so if we were to look at hydrogen on the periodic table, you would see hydrogen is represented by an H, and it would have a 1 at the top and a 1 at the bottom. This tells us there's 1 particle in the nucleus, and it tells us that 1 particle is a proton. There's a one proton. So if there's no neutrons, this atom does not have a neutron. If there's 1 positively charged particle, there has to be 1 negatively charged particle. So this 1 electron here that orbits around the first shell. So it has 1 electron and it has 1 valence electron. Over here we have carbon. Carbon is 6 for the atomic number, and it rounds to 12. So in the nucleus, we have 12 particles right here. 6 of them are protons. So therefore, 6 have to be neutrons. And if we have 6 protons, we have to have 6 electrons. We put 2 in the first shell. So therefore, we have 4 in the outer shell. And you can see they're evenly spaced. Where over here, it's kind of unevenly spaced, right? These are unevenly distributed, these valence electrons. Nitrogen, the symbol is N. We have 14 for the atomic mass and 7 for the atomic number. It's not always the case that the atomic number is half of the atomic mass. It's just the case in these 3 examples, but it's not always the case. So we have 14 particles of the nucleus, according to the atomic mass, and of those 14, 7 should be protons, right? And that's why it says 7 protons here. If there's 7 positively charged protons, there has to be 7 negatively charged electrons surrounding the outside, and that's why we see. This is another one where the electrons are scattered around that valence shell kind of unequally, right? Because there's 1, 2, 3, 4, there's 5 of them. If there were 4 of them, like in carbon, we could separate them evenly. That becomes important. All right, so to review, when we're looking at the periodic table, every atom or element is represented by a name, and every atom is represented by a symbol. And you can see the symbols don't always match the names, right? There's an atomic number, and there's an atomic mass. The atomic number is usually at the top on the periodic table, and the atomic mass is usually at the bottom when we're looking at the periodic table. All right. Now, if you're looking at a written report, like a lab report, they kind of reverse it, where you would represent the atomic number using a subscript, so it would be below, and you would represent the atomic mass as a superscript, so it would be written up above. But on the periodic table, carbon would be the symbol here, atomic number on top, atomic mass on the bottom. This tells us the number of particles in the nucleus. This tells us the number of protons, which it also happens to tell us the number of electrons, because that number is always the same. All right, so in nature we have these things called isotopes. Isotopes have the same number of protons but different numbers of neutrons. Let me show you an example. We looked at carbon. Carbon in nature has 6 protons with 12 particles in the nucleus. But also in nature, it can exist as a heavier form. Well, what does that mean? That means that in nature, we can have carbon with 6 protons, but with extra neutrons. So we have 13 particles in the nucleus. So we would still have 6 protons in the nucleus, but instead of 6 neutrons, we would have 7 neutrons. Another one is carbon-14. It doesn't change the number of protons, it just changes the number of neutrons. So these are called isotopes. The carbon that we're familiar with, that we find in our bodies, is carbon-12. But in nature, there are heavier versions of carbon that exist, and these are called isotopes. Isotopes are very unique because they are unstable. They break apart, they release energy. The release of this energy could be harmful, such as radiation, and if you're exposed to it, it can make you sick. But sometimes the release of this energy can be useful. We use carbon-14 to help us indicate the age of rocks. All right, so this radioactive material is found in rocks, and by determining the percentage of radioactive isotope carbon-14 in a rock, it tells us within a pretty good range, within 5,000 years or so, the age of that rock. And if we could determine the age of a rock, give or take 5,000 years, that's pretty good. That's pretty close, because 5,000 years, although it seems like a long time, is actually not a very long time when we kind of consider the geological timetable and how old this planet really is. If we could determine the age of rocks up to 5,000 years, that's pretty good. And why is that useful is because in the rocks, there are fossils. So by determining the age of the rocks, you could then determine the age of the organisms that are found in fossil form, embedded in those rocks. So this becomes a useful tool. So all of these elements are organized using the periodic table. The periodic table can seem very confusing when you look at it because there's a lot of information. But the periodic table does do a good job of organizing all the elements using a pattern that is pretty logical. All right. So right off the bat, you can tell within a minute of looking at the periodic table that all the elements are listed in order according to atomic numbers. All right. But then when we look at it more closely, there's also an organization in the periodic table that reveals how reactive the elements are. And what are they talking about here? That means that they are organized based on numbers of valence electrons. So the elements in a particular column, remember the columns go up and down. So all the elements in a column have the same number of valence electrons. They have the same number of electrons in their outer shell, right? So you can see that the atoms are arranged in order by atomic number going across. And then you'll get to the end, and it goes back to the beginning, the second row, 13 and 14, still organizing all the elements by the number of protons that are found in the nucleus. But if you look at the groups, at the columns, there's these numbers up here. 3, 4, 5, 6. And these numbers are at the top here, indicate the number. These are called group numbers. And this tells us the number of electrons. Not total, because that's what this number tells us, right? But this tells us the number of electrons in the outer shell, in the valence shell. So if I look at oxygen, which we already looked at, it has a total of 8 electrons. 2 of them are in the first orbital, in the inner orbital, but 6 of them remain in the outer one. Here's carbon. Carbon has 12 particles in the nucleus. 6 of those are protons. So it has 6 total electrons, but 4 of them, according to this number here at the top, 4 of them are found in the outer shell, in the valence shell. So the number of electrons in the outer shell kind of determine how reactive this atom is going to be. Right? And it tells us how likely these atoms are to create bonds with other atoms, right? And when we create bonds, we create compounds or molecules. So when we have atoms interacting with other ones, creating bonds, we create molecules. All right? There's a couple different ways that we could get these atoms to stick together. Okay? A couple different ways. We could create what are called ionic bonds, covalent bonds, and there's also these bonds called hydrogen bonds. If we look at the periodic table, all the way to the right, there's a column of elements. These column of elements are called the noble gases. And what makes them unique is their group number. If you look at the number all the way at the top of the column, the group number is 8. So these have 8 valence electrons. So their outermost shell is completely filled. Therefore, these atoms are extremely stable. They're happy, they're content. Their outer shell, their valence shell, is completely filled with electrons. These atoms are not likely to produce bonds with other atoms because they have a filled valence shell. It's all of the other atoms in the periodic table that have unfilled shells. So all the other ones have less than 8 electrons in their outer shell. And these atoms are more unhappy and are more eager to create chemical bonds with each other until they have those valence shells filled. All right. So there's a couple different techniques that atoms will use to fill those outer shells. If you're an atom and you have 1 electron in your outer shell, or 2 or 3, you're more likely to give up those 3 electrons to have an outer shell that's filled than you would to receive 7 or receive 6. And if you're an atom with 5, 6, or 7 electrons in the outer shell, you're more likely to accept an electron from somewhere else before giving up 7. All right. When we do this, when atoms do this, when they either give up electrons or receive extra electrons, they become what are called ions, and they take on a certain charge. When an atom is losing electrons, what happens is it's losing negative particles. It's donating negative particles. So as a result, it's going to form a positively charged ion. And when an atom receives extra electrons, it's receiving extra negative particles. And what that's going to do is that's going to result in the formation of a negatively charged ion. Right? And this is, we're kind of setting up the case for forming what are called ionic bonds. And let's look at sodium for an example. If we look at sodium and we count the number of electrons here, well, the first orbital only has 2. That's normal. The second one is completely filled with 8, so that's 10. But this last orbital has 1 electron in it, right? So it has 1 valence electron, totaling 11 electrons. Sodium has 11 electrons. Well, this is going to make sodium very unhappy because, like all the other atoms, it wants to have its outermost shell completely filled. So what sodium is going to be interested in doing is getting rid of this 1, donating that 1. And then what will be left is this shell underneath that is completely filled. And that will make sodium happy. So as it donates this negatively charged electron, it's losing a negative particle. And what happens when you lose a negative particle? So you have 10 electrons, but 11 protons. That gives you a charge of +1. And we represent that here by putting a plus sign. Now, if we look at chloride, it has the opposite problem. If we count the electrons in chloride, you'll see there's 2 in the first shell, 8 in the next shell. So that's 10, but 7 in the last shell. This makes chloride very unhappy because it was so close to filling the shell. It just needs 1 more. So what it'll do is it'll look for a free electron. Just like how sodium gave this 1 up, chloride would be interested in accepting that. And when it does, it puts it in that outer shell, and it makes that outer shell, that valence shell, filled. But now it's receiving an extra negative particle. So when we have 17 protons, now we have 18 electrons, and this causes the atom to create a negatively charged ion, giving this a charge of -1. Well, do you know what happens when we have atoms that are negative coming in close contact with other atoms that are positive? As you can guess, opposite charges attract to each other. The positively charged sodium ion and the negatively charged chloride ions are attracted to each other, and they create what's called an ionic bond due to this opposite attraction, creating a molecule of sodium chloride, which is salt. All right? So this is a very common type of bonding that results from gaining or losing electrons. All right, so just remember, when we're talking about ionic bonds, the most important thing is that this results from either gaining or losing electrons. And what that does, when you gain an electron, you become an ion, a negatively charged ion. And when you lose an electron, like sodium does, you become a positively charged ion. And then you are therefore attracted to it, one another. But both of these atoms are happy because their valence shells have been filled. All right, so that's ionic bonds. That's not the only way to form bonds. There's another way, and this is called covalent bonds. Covalent bonds work a little bit differently. Covalent bonds are the strongest bonds. It's important to have strong bonds making up the molecules of the body because we don't want the molecules to fall apart, right? But as important as it is to have strong bonds making up many of the molecules, it's also important to have some weak bonds because there are molecules that we do want to have come apart in the right places. And the one molecule that comes to mind that needs to come apart very often is DNA. You know, every time we make new cells, we have to copy the DNA. In order to copy the DNA, we have to get it to come apart. We make copies of each side because DNA is a double-sided molecule. So getting it to come apart requires the fact that it's held together in the first place with weak bonds. But we want it to come apart in the right spot. We don't want to break it apart on the sides. We want it to unzip up the middle. So the sides are held together by strong covalent bonds. And these covalent bonds are produced by not by donating and giving electrons away or receiving electrons, like we saw in ionic bonds, but more of from like sharing electrons. There's instances where these atoms will share electrons. And let me show you an example. This is an atom of oxygen here, right? So oxygen, if we count the electrons in oxygen, there should be 1, 2, 3, 4, 5, 6, 7, 8. All right, so oxygen has 8 total electrons, but only 6 in the outer shell. So this makes oxygen kind of unhappy. Because remember, like all the other atoms, it wants to have a filled outer shell, so it needs 2 more. Well, hydrogen, if you remember, only has 1 electron in its outer shell. Remember, the first shell can only hold 2. Hydrogen only has 1 shell, so it's only looking for 1 more, right? So what hydrogen can do is it could kind of come over here and become part of this outer shell here on this oxygen. So it'll kind of take its 1 electron and join the party here. So now it has 2 electrons in its outer shell because it's going to share one of these electrons with this oxygen. And this hydrogen atom could do the same thing. It could go over here and share this electron from that is one of oxygen's. And now this hydrogen has 2 electrons in its outer shell. This one has 2 because they're going to share. And the oxygen now has 8. But what happens is, because of this unequal distribution of electrons around in the valence shell, what happens is, even though these 3 atoms are sharing the electrons, the sharing becomes unequal, right? So there's a lot more mass here in this nucleus. Remember, there's 16 particles in that nucleus. There's only 1 particle in that nucleus and 1 particle in that nucleus. So there's more mass around here. So these electrons are more likely to kind of hang out around this oxygen. But every so often, the electrons will orbit these hydrogen atoms as well. But because the negatively charged electrons are hanging out more so on this end and not so much on this end, there's a electromagnetic shift inside of this molecule. So this end of the molecule takes on a slightly negative charge compared to this side, which is taking on a slightly positive charge. When this happens, and of course, when we put two hydrogens on an oxygen molecule, we create water. This is H2O. Water, because of this unequal distribution or unequal sharing of electrons, water is considered a polar, or is considered to be held together by what are called polar covalent bonds. There's sharing of electrons that is happening, but this sharing of electrons is not equal sharing. The oxygen's kind of hogging the electrons for itself, and only sharing with these hydrogens very little, right? But nevertheless, each of these is happy because their valence electrons are filled. Now, because the oxygen end is keeping the electrons more, electrons are negatively charged. Therefore, this end takes on a slightly negative charge, and this end takes on a slightly positive charge. Let me show you an example of a covalent bond. I'm just looking for room to write. Let's say we have carbon. I'm going to draw carbon over here. Remember, carbon has 12 particles in the nucleus. In the first shell, there's 2 electrons. In the second shell, there's 4. 1, 2, 3, 4. Because there's 4 in the valence shell, they're equally distributed. So if carbon was to do the same thing and share electrons with 4 hydrogens, that would look like this. Oop. Now carbon becomes very happy because now it has its valence shell completely filled. Hydrogens become happy. Their valence shells are completely filled. They can only have 2 on their outermost shell because they only have 1 shell. And we create what's called CH4. This is called methane gas. But what's different about this is the sharing is equal, right? So the electrons are orbiting the carbon and each hydrogen equally. So this is called a nonpolar covalent bond. Because the electrons are being shared equally amongst all the different components, it doesn't take on a slightly negative charge on one end and a slightly positive charge on the other end like water does. All right? So this becomes useful, though, however, having polar molecules, because when we have a water molecule that has a positive charge on one end and a negative charge on the other, positive charge over here and a negative charge over here, positive over here, negative over here, negative over here, positive over here. What happens is these water molecules become attracted to each other. The positive ends of one water molecule will be attracted to the negative ends of another, and we can get these water molecules to kind of cling to each other. All right. Now this sets us up for the third type of bonds, which are called hydrogen bonds. And hydrogen bonds form as a result of these weak attractions that exist between adjacent molecules that are polar. And it's these hydrogen bonds that cause these water molecules to kind of cling together. It's hydrogen bonds that cause DNA strands to cling together. And it's these hydrogen bonds that form some ends of proteins to want to be attracted towards one another, helping to maintain the shape of proteins. All right, moving on. The forces that exist between these resonating molecules, this attractive forces, these are called van der Waals forces. All right, so the resulting, the forces that result from intermittent electromagnetic interactions between resonating molecules, like we see between these water molecules. These are forming hydrogen bonds, but through these forces called van der Waals forces. Sometimes bonds contain energy. We store energy in bonds, right? And we could release that energy when we break the bond. When energy is released as a result of breaking a bond, we produce what's called an exothermic reaction. Exo means out, right? So energy is being released. Some bonds absorb energy. When energy is stored in the bonds, when we put bonds together, we create what are called endothermic reactions. Remember, endo means in. So we're putting energy into the bonds. We're storing them to be used for later on. So when we eat our food, sometimes we take these glucose molecules, which are really small, and the liver will tie them together into long chains, right? Long chains of sugars that we would store in the form of glycogen. And we would use covalent bonds to store these glucose molecules in these long chains. And the idea is that when we bond these glucose molecules together, we're doing that because we're storing the energy in these bonds. We use sugars for energy. This is just a visual interpretation of photosynthesis. This is photosynthesis here. Photo means light. And synthesis means to produce or to make something or to build. So photosynthesis literally means making from light. But the plant not only uses light to make glucose, but it also uses carbon dioxide. And it also uses water. We need water for this to happen. So water, carbon dioxide combined with sunlight allows this plant cell in the chloroplast to put these atoms together using covalent bonds, making a molecule of glucose. And glucose is going to be, well, it's a food nutrient. And the idea is when we produce these covalent bonds, there's energy that is stored in these bonds. And the energy can be released when those bonds are broken. Water is considered life's essential chemical. It is the most abundant molecule in all living things, makes about 60 to 70% of our total body weight. And we need water specifically in our cells, right? In the cytoplasm of the cells to carry out cellular metabolic functions, but we need it in our bodies too to carry out very important chemical reactions that are happening for digestion, also respiration. We need it in our blood. It's a transport medium. It makes up most of our blood. All right. So without an adequate supply of water, we wouldn't be able to perform many of the chemical reactions that we need in order to live. And so our cells would no longer function. And because our bodies are made up of collections of cells, that would mean that our bodies would no longer function. And when we look at water, there are 6 important properties of water. First one being that the water is liquid at room temperature. This is very important because, as you know, water can go from a liquid to a vapor or from a liquid to a solid. And it's very important that we keep water in its liquid form, especially when it's occupying our bodies, right? All right. So water is liquid. It's the universal solvent. it's able to dissolve other substances. All right, so that's also very important. Hydrophilic substances are substances that immediately become separated in water, like salt. If it can dissolve in water, it's considered hydrophilic. Hydrophobic substances are not soluble in water. This would be things like oils or fats. These are hydrophobic. All right? They repel water. It's also very important. Water is cohesive and adhesive, right? It allows the cohesive and adhesive properties of water allows it to fill our cavities, right? Whether it's a blood vessel or any of our membrane-bound cavities or cells, membranes. It provides lubrication, right? And it provides an excellent transport medium. Like I said, most of your blood, a lot of what's your blood comes in the form of red blood cells, but more than half of the blood is just water, right? Coming in the form of plasma. And this plasma is what's moving all the other cells of the blood around. It has a high specific heat. This is important because what that means is it takes a lot of energy to change the temperature of water. If you think about the temperature outside, last night, the temperature outside was probably in the 50s. It was about 58 degrees last night. And today, the temperature is going to be close to 80 degrees. So that is a temperature difference of 22 degrees, right? 58 to 80 degrees. Within 24 hours, the air temperature can change up to 22 degrees. Now, water, if you were to go outside and take the temperature of the pond that's close to your house, it might read a certain temperature. Let's say it reads 75 degrees. And in 24 hours, it might drop 1 degree, maybe 2 degrees. And the reason for that is because it has a high specific heat. It takes a lot of energy to change the temperature of water. It's a very stable molecule, right? And that's good because we want our bodies to maintain a certain temperature. And the fact that our bodies are filled with water, that only helps to maintain our body's temperature, keeping it stable. It takes actually the amount of energy that is required to change the temperature of water. One gram of water or 1 milliliter of water by 1 degree is considered 1 calorie. Right? That's how we measure calories. Right. So this keeps the body temperature stable. It has a high heat of vaporization, so it requires a lot of energy to convert water from its liquid form into a vaporized form. Right. Another unique property of water is that when it goes into a solid form, its density decreases because what happens is air gets trapped in it, and the volume expands. So volume is increasing, but mass is not. So whenever we have a density calculation where the volume is higher than the mass, well, then the density calculation is one that calculates to be less than 1. When the density value is calculated to be less than 1, well, that object is going to float. If the density calculation computes to be greater than 1, then that object will float, will sink. Excuse me. All right. So, water, in its normal form, its liquid form, water has a mass of 1 gram per 1 milliliter. So the density of water in liquid form is 1.0 grams per milliliter. But when it freezes, the volume, this unit down here, becomes larger. And so the density value drops below 1. So ice floats. Now, pH kind of has something to do with water, right? So if we look at a water molecule, we have an oxygen, we have a hydrogen, We have another hydrogen over here. If we were able to break this apart, let's say we broke it here, what would we be left with? Well, we would be left with a hydrogen atom and a hydroxide atom, like this one. Now, there are certain things in the world that have more of these in them, and there's other things in the world that have more of these in them, right? Some solutions that have more of these in them these are called acids. And other solutions that have more hydroxide ions in them, these are called bases. And you could measure the concentration of hydrogen atoms or hydroxide atoms, and we could create what's called a pH scale. The pH scale has a dividing line in the middle. This is called neutral. So 7 is a neutral pH. That means for every one of these, there's one of these. And a great example of something that has one of these, for every one of these, is water, right? We saw this. Water, this is how we started. Water has a pH of 7 as a neutral pH. It's not an acid or a base. But things like coffee, beer, wine, battery acid, all these different things contain different concentrations of hydrogen ions, and they get stronger and stronger as we go to each end of the scale. So these things over here, these are called weak acids. These things down here at the end of the scale, these are strong acids. Same thing over here. If we go just above 7, these are weak bases. But down here, bleach would be down here. Ammonia would be down there. These are strong bases. Right? So this is just a scale. Our blood is somewhere in here. It's on the slightly basic side of neutral, the blood. In fact, this is one of the things that your body does to maintain homeostasis, to keep your blood pH within a certain range. Blood pH should be somewhere between 7.35 and 7.45. Right? And because carbon dioxide is an acid, our breathing helps to regulate the blood pH. Right? So if our blood pH is becoming acidic for whatever reason, we'll just breathe more, breathe heavier, getting rid of more carbon dioxide gas, and that'll fix it. The lungs are very important in maintaining the blood pH. All right, so your notes say that your blood pH is somewhere between 7.4 and 7.5, but actually it's, to get exact, it's 7.35 to 7.45. Right? If you breathe slowly, let's say you come across somebody that has been shooting heroin or something, and they're passed out, and their breathing is slowing down. So what's going to happen is carbon dioxide levels are going to start to rise in the body. And remember, carbon dioxide is an acid. So that's going to make the blood become more acidic. So in order to fix this, because this will become a problem, this is called blood acidosis. This could cause the person to go into a state of shock. You would need to start breathing for that person in order to adjust the blood pH. Now, the kidneys also will play a part in what's called metabolic stabilization. They're going to start to release different buffers. When the blood is becoming too basic, it'll release these hydrogen ions. But when it becomes too acidic, it'll release bicarbonate ions to correct the problem. So even though there's metabolic compensation happening by the kidneys, it's really the lungs that are responsible for maintaining proper blood pH. All right. Okay, like I said in the beginning, the body is composed of organic compounds. Organic compounds. All right. The major organic compounds that we find in our bodies, in the bodies of all living things, are going to be carbohydrates, lipids, proteins, and nucleic acids. Nucleic acids is what puts the Na in DNA. All right? And that's our genetic material. Carbohydrates are sugars, right? So they're extremely abundant. We get them when we eat foods. And if we look at all the carbohydrates, for example, glucose has 6 carbons, 12 hydrogens, 6 oxygens. So the formula is C6H12O6. And when we combine these glucose molecules, um, we, we combine them in these chains. When plants combine glucose molecules, they make long and straight chains. This is cellulose. Cellulose is a carbohydrate that's found in cell walls. And this is a very strong carbohydrate. But glycogen is how we store glucose molecules in the liver. And it's a little bit different because it's more branchy. Glycogen is an animal product. Cellulose and starch, these are plant products. Cellulose is actually too strong for our bodies to break down. So when we eat lettuce, for example, we're eating all this cell wall and whatnot, but we don't get any nutritional value from it because we can't break it down. Our digestive systems can't break that down. Where we can break down starch and we can break down, the glucose molecules that we form in our livers in the form of glycogen. So when we have single unit sugars, like glucose is a 6-sided single unit, this is called a mono. Mono means one. Saccharide. Saccharides are sugars. So single unit sugars, glucose is the most common example, but also fructose, ribose, deoxyribose, maltose. We have monosaccharides. So mono is one sugar. Disaccharides is when we get 2 units of sugar bonded together. So we have a monosaccharide here, glucose, and another monosaccharide over here, glucose. And we form what's called a disaccharide. When we've put glucose together with fructose, we get sucrose. Sucrose is a disaccharide. That's table sugar. Lactose, the sugar that's in milk, is a disaccharide. It's formed when we combine glucose with galactose. All right. And then we get long chains when we combine many, many, many individual carbohydrates together. We get long chains. Remember, glycogen is how we store glucose in our livers, right? So this is an animal product. Starch and cellulose, these are plant products. Starch is stored glucose, usually found in the roots, right? In the potatoes or in tubers and whatnot. But cellulose is a little bit different because it's the way that the molecules are arranged. It's for strength, really, right? Making up the cell walls. Lipids, on the other hand, are not water soluble, right? So we call hydrophobic. They don't dissolve in water. They don't break apart in water. In fact, they repel water. Right? The lipids that we store in our bodies are coming in the form of fatty acids. And if we look at fatty acids, we see commonly these things called triglycerides. Tri means that they have 3 units coming off of the backbone. The backbone is called a glycerol. So let's say we have a glycerol here. And the glycerol is just made up of 3 carbons with oxygens and hydrogens poking of it. Then we would have these long chains, 3 of them, of carbon and hydrogen, repeating, very long chains. So when we have 1, 2, 3 really long chains of hydrocarbons on this glycerol backbone, this is what a triglyceride is. Phospholipids are a little bit different. We'll take a look at phospholipids, but phospholipids are the types of lipids that we find in our cell membranes. When we look at these long chains of fatty acids, they kind of come in two different forms. They're either unsaturated or saturated. Well, let's look at that. Over here, if our glycerol was up here, remember we could have 3 chains. Well, look at this chain. This is one chain. If we look at all the carbons here, we could see that each carbon has 2 hydrogens surrounding it. So all these carbons have 4 hands, right? Remember, there's the abilities for a carbon to make 4 bonds. Remember that. So this carbon is making bonds with the 2 neighboring carbons, and then with its 2 leftover bonding sites, we have hydrogens. So every carbon is completely saturated. We cannot squeeze any more hydrogens in here at all. It's completely filled. Therefore, these fatty acids are called saturated fats or saturated hydrocarbon chains. Now, if we look over here, we see 2 carbons that instead of holding on to hydrogens are holding on to each other using 2 bonding sites. So this is an unsaturated hydrocarbon chain. Whenever we have an unsaturated chain, and we see it happening right here, it causes a kink in the fatty acid chain. All right, so when we see this, we have unsaturated fats. The unsaturated fats are usually liquid at room temperature. So oil is an unsaturated fat. And the saturated fats are usually solid, like butter, at room temperature. This is a saturated fat, or it's just actually a saturated hydrocarbon fatty acid chain. With phospholipids, there's 2 parts to a phospholipid. There's a phosphate head, and then there's these lipid tails, 2 of them. And they kind of arrange themselves in rows. These phosphate heads do. Like this. There could be multiple layers of them, right? All right, so each phosphate head has these has 2 lipid tails associated with them. So this is called a phospholipid bilayer. And why is it called a bilayer? Because there's 2, bi means 2 layers. We have this layer and we have this layer. So this, what I'm drawing, is a small segment of what's called a cell membrane. So on one side of the cell membrane, there's going to be water on the outside of the cell. And on the other side of the cell, there's going to be water on the inside of the cell. This is called cytoplasm, right? So what separates the inside of the cell from the outside of the cell is this cell membrane. The cell membrane is made up of phospholipids. The phosphates heads, these are hydrophilic, meaning that they like the water, which is why they're in contact with the water. So these phosphate heads down here are hydrophilic also, and they come in contact with the water inside the cell. Now, the lipid tails are hydrophobic, meaning that they do not want to come in contact with any water. So they face each other, and this area is water-free. There would be no water in here. This would repel water. So what this does is it's these hydrophobic and hydrophilic properties of the phospholipid bilayer is what maintains the shape of the cell membrane. And now, this is not a solid structure. It's more of a fluid structure. And what happens is, as things go through the cell membrane, the phospholipids can become separated. But because of the properties, they just find each other and come back together. So this is a good picture of the phospholipid bilayer here, of the cell membrane. Hydrophobic here. These are the hydrophobic fatty tails, and these are the hydrophilic phosphate heads, right? Hydrophilic. This is one, what's called phospholipid. Then we have these other types of lipids, steroids and cholesterol, that we find the sex hormones. And we find that they look much different. They don't really have these long hydrocarbon tails. They come usually in the form of rings, but they're lipids nevertheless. So this is some visual interpretation. Cholesterol, you can see that they arrange themselves in these rings, not really these long tails like triglycerides. Now with proteins are found all over the body, right? All of your structures are made from proteins. And when we look at proteins, they have subunits. The subunit of a protein are amino acids. Okay, so if I want to build a house, a log cabin, the log cabin is the structure. The subunits would be logs, right? You put the logs together to make the log cabin. Well, if I want to put proteins together, I got to put amino acids. I got to combine amino acids. And there's 20 different amino acids that are available in the world, right? In your bodies. And how are they all different from each other? Well, how they're all the same is that they have this central carbon with a hydrogen, with a nitrogen, creating an amino group. They all have that. And then they have a carbon with this carboxyl group with these two oxygens here. So they all have that. Now, what makes them different is what's coming off of here. This is called the side chain. And there's 20 varieties of these different side chains. And the side chains is what gives the amino acids their unique properties, right? So we could see down here, we're showing you just 4 of the 20. So you can see this amino acid has an amino group, a carboxyl group, a hydrogen. And for the R group, this is the most simple one. It's just one hydrogen. This is glycine. But you can see they can become more and more complex, how they vary. This is called the variable group or the R side chain. And remember, there's 20 different varieties. But when we put proteins together using 20 different amino acids, well, you could form an unlimited amount of different proteins. All right. So as we combine amino acids together, we form what are called polypeptides. Poly means many amino acids. And these polypeptides, as they develop, will take on unique shapes and become more and more complex and eventually develop into what are called proteins as they become more three-dimensional. All right. And the shape is very important. And the shape is determined by the types of amino acids that we use. But the shape is very important because it determines the function. The shape of the protein is determined by the function of the protein. So what are some shapes? Well, when we start putting amino acids together, we have what are called, this is an amino acid chain, right? We have one amino acid here, another one here, another one here. So we're making an amino acid chain. Well, we would refer to this as a primary structure, all right? But as we start to really pile them on one after another, different shapes can start to occur. A common shape is this helix shape. It twists around forming a helix. This is a common shape. Or it could start to fold like a folded piece of paper like this. This is called beta pleated sheets. There's a third one that's just random coiling, right? So alpha helix, beta pleated sheets, random coils. These are all pretty common shapes that can occur as we start to really pile on amino acids. But as we start to combine alpha helix with some pleated sheets, with some random coils, we get into what are called tertiary structures. And when we have multiple tertiary structures combined with other tertiary structures, we get these really advanced or elaborate shapes of polypeptide chains. And this is where we get to what is called a protein. And it's this unique shape here, right, of the protein that allows this protein to perform its specific function. All right. Remember, if a protein loses its function, it's probably because it lost its shape. All right. So the important thing to know here is that function comes from shape. And there's many, many different types of shapes. And the shapes come from the amino acids that are being used. Because all those amino acids have very specific characteristics about them. All right, so what happens? Well, if we change even one amino acid, it can alter the shape of the whole protein, and it could devastate the protein's function. This is what's happening in sickle cell anemia. There's a mutation in the DNA. It causes one amino acid out of many, many thousands of amino acids that are used to form the protein hemoglobin. And it causes serious problems. All right. So this is just one little tiny piece of a really big protein that has devastating effects on the functioning of a protein. All right, some proteins work as what are called enzymes. An enzyme is a specific protein that speeds up a specific chemical reaction. You have enzymes in your mouth. You have enzymes in your stomach. These are digestive enzymes. And what they do is they grab onto food nutrients and break them apart quickly. All right. In order for these enzymes to work, they have to be the correct shape. Because remember, enzymes are proteins, and the function of a protein is highly dependent on the shape of the protein. So there is an active site where the enzyme grabs onto the food substance, and it starts to perform its job, right? It starts to perform its digestive function. If for some reason the enzyme loses its shape, it will lose its function. So here we see an enzyme. And it looks like this is a substrate. It looks like this is a disaccharide, right? So it looks like a carbohydrate made up of two units, a disaccharide. So this disaccharide would fit into this active site of the enzyme, and then the enzyme would perform its function, and then it release the products. The products would be released as monosaccharides that the cells can absorb and use for energy, or be sent to the liver where we can store it in the form of glycogen, so we can have that energy later. All right, so as long as this enzyme is maintaining its shape, this enzyme will be recycled and will perform this function over and over and over again. But let's say there is a disruption of homeostasis where the body temperature increases. Right? An increase of temperature could cause these enzymes to lose their shape, to become denatured. Or let's say for some reason you're going into blood acidosis, and the pH of the body is being altered. Well, an alteration of pH could also cause these proteins to lose their shape and become denatured. All right. So two ways to really mess up proteins. Number one is a change in temperature, and number two, a change in pH. If we maintain this, typically we can maintain our enzymes. But if your temperature gets above, let's say, 105 degrees, it could have devastating effects, because your enzymes or your proteins will start to become denatured, and that will have result in deadly consequences. All right. So when it comes to nucleic acids, remember, nucleic acids, this is the Na that we see in DNA and RNA. DNA is deoxyribonucleic acid. And we find DNA in the nucleus of our cells, right? And the nucleus is where DNA is always kept, right? That's where your hereditary information. So what is DNA? DNA is a code for making proteins. It's a code for making proteins. We use this code to build proteins and to maintain homeostasis. Now, RNA is also built to the nucleus, but it's able to leave the nucleus because that code has to hook up with a ribosome. So the ribosomes could read the code and build the proteins. DNA cannot leave the nucleus, it's too valuable. All right. So the DNA is a genetic code, right? It's made up of these units here. These are called nitrogen bases. There's 4 varieties of nitrogen bases. There's phosphates and there's sugars. Phosphate, sugar. Each phosphate is connected to a sugar, and each sugar is connected to one of the 4 varieties of nitrogen bases. Now, there's A's for adenine, C's, G's for guanines, and T's for thymines. A's always pair with T's, and C's always pair with G's. And it's these codes that come in the form of A's, C's, T's, and G's that provide us with our genetic code. All right. It says here life requires energy. And we have high energy compounds that we use to power the cells, right? And as a result, power our bodies. Remember, when you're eating a lot of food, let's say you eat a lot of pasta, you'll use some of that sugar that's in the pasta for short-term energy, right? So you'll send some of those um, molecules, those glucose molecules, to your cells, and we'll use that for short-term energy. But there comes a time where we have too much sugar in the blood, and we want to take some of that sugar and get it out of the blood. And we could store that sugar in our livers and in our muscles, and we could use that liver later on. When we store that sugar, we put it into chains. We combine those molecules into long chains in the liver. Those chains are called glycogen. We could also store triglycerides in our bodies in these specialized cells called adipocytes. These are fat cells. And we store fat in our bodies, and we store sugar in our liver and our muscles to use for later. So this is a long-term energy supply, right? Glycogen and fats, these are long-term energy nutrients, where simple carbohydrates are more of a short-term energy supply. ATP is an energy molecule that's made in the mitochondria of the cells. And pretty much what it is adenosine triphosphate. So we have its adenine group here. We have a sugar portion. And then we have 1, 2, 3 phosphates. When we combine these phosphates, there's energy that is being stored in these covalent bonds. Right? So we produce ATP, adenosine triphosphate, by combining these phosphate molecules. Energy is stored there. When we make ATP in the cells at the mitochondria, this is called cell respiration, right? So we would need food nutrients, we would need oxygen to get this to happen. Now, when we want to use the energy, what we do is we take this ATP molecule that has 1, 2, 3 phosphates, and we break this phosphate off as a result, breaking this bond right here. When we break this bond right here, we produce an exothermic reaction that releases energy. And then as a result of cell respiration, this ADP, this becomes ADP, adenosine diphosphate, goes through the process of cell respiration, and we put the 3rd phosphate back on as a result of what's called cell respiration. So this is a constant thing that's happening in all of your cells. You're breathing in oxygen, you're eating food, and those things get sent to your cells, and the mitochondria use those 2 things to produce ATP. So ATP, adenosine triphosphate, is our energy molecule. All right. And this is the end of the note. So what I recommend you do, go to the textbook, right? Where's that textbook? Week 1. Week 1 has all the information, right? So here's the textbook. Open up that textbook. Read the information in the book. I know it seems that you don't have a lot of graded assignments, but you do have plenty to do, right? Chemistry of life. All right, so chemistry, bonds, covalent bonds, ionic bonds, hydrogen bonds. We looked at water, macromolecules. This is proteins, lipids, carbohydrates, right? Going from DNA to a protein, there's more specific information about that. But the concept check is where you should be spending time, right? Look at these questions, listen, explain the importance of water, right? There were several slides that we talked about water. Compare and contrast acids and bases, right? What is the full of phospholipids? Well, we already talked about that. They make cell membranes, right? What is ATP? That's the last slide. But look in the book and see what it says about these things. Get a good idea of how to answer these questions because you will see these questions coming up. All right. And so be prepared for that. All right. If you have any questions on this, feel free to email me. And there are no grade assignments due this week. But I hope you enjoy the rest of your week and stay tuned for the next lab. That'll be posted shortly. All right. Bye bye.